Protein C, also known as autoprothrombin IIA and blood coagulation factor 14, is a zymogen, the activated form of which plays an important role in regulating anticoagulation, inflammation, cell death, and maintaining the permeability of blood vessel walls in humans and other animals. Activated protein C performs these operations primarily by proteolytically inactivating proteins factor VIIIa and factor VIIIa. APC is classified as a serine protease as it contains a residue of serine in its active site. In humans, protein C is encoded by the PROC gene, which is found on chromosome 2. The zymogenic form of protein C is a vitamin K-dependent glycoprotein that circulates in blood plasma. Its structure is that of a two-chain polypeptide consisting of a light chain and a heavy chain connected by a disulfide bond. The protein C zymogen is activated when it binds to thrombin, another protein heavily involved in coagulation, and protein C's activation is greatly promoted by the presence of thrombomodulin and endothelial protein C receptors. Because of EPCR's role, activated protein C is found primarily near endothelial cells, and it is these cells and leukocytes that APC affects. Because of the crucial role that protein C plays as an anticoagulant, those with deficiencies in protein C, or some kind of resistance to APC, suffer from a significantly increased risk of forming dangerous blood clots. Research into the clinical use of activated protein C, also known as drotocoagulant alpha activated, has been surrounded by controversy. The manufacturer Eli Lilly and Company ran an aggressive marketing campaign to promote its use in people with severe sepsis and septic shock, including the sponsoring of the 2004 Surviving Sepsis Campaign guidelines. A 2011 Cochrane review, however, found that its use cannot be recommended as it does not improve survival. History: protein C's anticoagulant role in the human body was first noted by Seegers et al. In 1960. Who gave protein C its original name, autoprothrombin 2A. Protein C was first isolated by Johann Stenflo from bovine plasma in 1976, and Stenflo determined it to be a vitamin K-dependent protein. He named it protein C because it was the third protein that eluted from a DEA-Ceph-Rose ion exchange chromatograph. Seegers was, at the time, searching for vitamin K-dependent coagulation factors undetected by clotting assays, which measure global clotting function. Soon after this, Seegers recognized Stenflo's discovery was identical with his own. Activated protein C was discovered later that year, and in 1977 it was first recognized that APC inactivates factor VIIIa. In 1980, VIIIa and Davy discovered that APC also inactivates factor VIIIa, and soon after, protein S was recognized as a cofactor by Walker. In 1982, a family study by Griffin et al. First associated protein C deficiency with symptoms of venous thromboses. Homozygous protein C deficiency and the consequent serious health effects were described in 1984 by several scientists. cDNA cloning of protein C was first performed in 1984 by Beckman et al., which produced a map of the gene responsible for producing protein C in the liver. In 1987 a seminal experiment was performed whereby it was demonstrated that activated protein C prevented coagulopathy and death in baboons infused with lethal concentrations of E. coli. In 1993, a heritable resistance to APC was detected by Dalbecurrent CCKAL and associated with familial thrombophilia. In 1994, the relatively common genetic mutation that produces factor Gliadin was noted. 2 years later, gliadomainless APC was imaged at a resolution of 2.8 AANGSTR. A paragraph was beginning with the PROC clinical trial of 2001. It was recognized that many of the symptoms of sepsis may be ameliorated by infusion of APC, and mortality rates of septic patients may be significantly decreased. Near the end of that year, drotocoagulant alpha, a recombinant human activated protein C, became the first drug approved by the US FDA for treating severe sepsis. In 2002, Science published an article that first showed protein C activates protease-activated receptor 1, and this process accounts for the protein's modulation of the immune system. Synthesis, structure and activation. Human protein C is a vitamin K-dependent glycoprotein structurally similar to other vitamin K-dependent proteins affecting blood clotting, such as prothrombin, factor VII, factor IX and factor X. Protein C synthesis occurs in the liver and begins with a single-chain precursor molecule, a 32-amino acid N-terminus signal peptide preceding a propeptide. Protein C is formed when a dipeptide of Ls198 and Arg199 is removed. This causes the transformation into a heterodimer with N-linked carbohydrates on each chain. The protein has one light chain and one heavy chain connected by a disulfide bond between Cis183 and Cis319. Inactive protein C comprises 419 amino acids in multiple domains, 1 GLA domain, a helical aromatic segment, 2 epidermal growth factor-like domains, an activation peptide, and a trypsin-like serine protease domain. The light chain contains the GLA and EGF-like domains and the aromatic segment. The heavy chain contains the protease domain and the activation peptide. It is in this form that 85–90% of protein C circulates in the plasma as a zymogen, waiting to be activated. The remaining protein C zymogen comprises slightly modified forms of the protein. Activation of the enzyme occurs when a thrombin molecule cleaves away the activation peptide from the N-terminus of the heavy chain. The active site contains a catalytic triad typical of serine proteases. The activation of protein C is strongly promoted by thrombomodulin and endothelial protein C receptor, the latter of which is found primarily on endothelial cells. The presence of thrombomodulin accelerates activation by several orders of magnitude, and EPCR speeds up activation by a factor of 20. If either of these two proteins is absent in marine specimens, the mouse dies from excessive blood clotting while still in an embryonic state. On the endothelium, APC performs a major role in regulating blood clotting, inflammation, and cell death. Because of the accelerating effect of thrombomodulin on the activation of protein C, the protein may be said to be activated not by thrombin but the thrombin-thrombomodulin complex. Once in active form, APC may or may not remain bound to EPCR, to which it has approximately the same affinity as the protein zymogen. The GLA domain is particularly useful for binding to negatively charged phospholipids for anticoagulation and to EPCR for cytoprotection. One particular exercit augments protein C's ability to inactive factor VIIIa efficiently. Another is necessary for interacting with thrombomodulin. The biologic instructions for synthesizing protein C in humans are encoded in the gene officially named protein C. The gene symbol approved by the Hugo Gene Nomenclature Committee is PROC from protein C. It is located on the second chromosome and comprises 9 exons. The nucleotide sequence that codes for human protein C is approximately 11,000 bases long. Protein C in zymogen form is present in normal adult human blood plasma at concentrations between 65–135 IUDL. Activated protein C is found at levels approximately 2,000 times lower than this. Mild protein C deficiency corresponds to plasma levels above 20 IUDL, but below the normal range. Moderately severe deficiencies describe blood concentrations between 1 and 20 IUDL. Severe deficiencies yield levels of protein C that are below 1 IUDL or are undetectable. Protein C levels in a healthy term infant average 40 IUDL. The concentration of protein C increases until 6 months, when the mean level is 60 IUDL. The level stays low through childhood until it reaches adult levels after adolescence. The half-life of activated protein C is around 15 minutes. Pathways. The protein C pathways are the specific chemical reactions that control the level of expression of APC and its activity in the body. Protein C is pleiotropic, with 2 main classes of functions, anticoagulation and cytoprotection. Which function protein C performs depends on whether or not APC remains bound to EPC after it is activated. The anticoagulative effects of APC occur when it does not. In this case, protein C functions as an anticoagulant by irreversibly proteolytically inactivating factor VIIa and factor VIIa, turning them into factor VI and factor VI respectively. When still bound to EPCR, activated protein C performs its cytoprotective effects, acting on the effector substrate PAR1, protease-activated receptor 1. To a degree, APC's anticoagulant properties are independent of its cytoprotective ones, in that expression of one pathway is not affected by the existence of the other. The activity of protein C may be downregulated by reducing the amount either of available thrombomodulin or of EPCR. This may be done by inflammatory cytokines, such as interleukin-1I² and tumor necrosis factor I+, or minus. Activated leukocytes release these inflammatory mediators during inflammation, inhibiting the creation of both thrombomodulin and EPCR, and inducing their shedding from the endothelial surface. Both of these actions downregulate protein C activation. Thrombin itself may also have an effect on the levels of EPCR. In addition, proteins released from cells can impede protein C activation, for example eosinophil, which may explain thrombosis in hypereosinophilic heart disease. Protein C may be upregulated by platelet factor 4. This cytokine is conjectured to improve activation of protein C by forming an electrostatic bridge from protein C's GLA domain to the glycosaminoglycan domain of thrombomodulin, reducing the Michaelis constant for their reaction. In addition, protein C is inhibited by protein C inhibitor. Anticoagulative effects. Protein C is a major component in anticoagulation in the human body. It acts as a serine protease zymogen. APC proteolyzes peptide bonds inactivated factor V and factor VIII, and one of the amino acids in the bond is serine. These proteins that APC inactivates, factor VA and factor VIIa, are highly procoagulant cofactors in the generation of thrombin, which is a crucial element in blood clotting. Together they are part of the prothrombinase complex. Cofactors in the inactivation of factor VA and factor VIIa include protein S, factor V, high-density lipoprotein, anionic phospholipids and glycosphingolipids. Factor VA binds to prothrombin and factor ZA, increasing the rate at which thrombin is produced by 4 orders of magnitude. Inactivation of factor VA thus practically halts the production of thrombin. Factor VIII, on the other hand, is a cofactor in production of activated factor X, which in turn converts prothrombin into thrombin. Factor VI augments factor X activation by a factor of around 200,000. Because of its importance in clotting, factor VIII is also known as anti-hemophilic factor, and deficiencies of factor VIII cause hemophilia A. APC inactivates factor VA by making 3 cleavages. The cleavages at both ARG306 and ARG506 diminish the molecule's attraction to factor ZA, and though the first of these sites is slow to be cleaved, it is entirely necessary to the functioning of factor V. Protein S aids this process by catalyzing the proteolysis at ARG306, in which the A2 domain of factor V is dissociated from the rest of the protein. Protein S also binds to factor ZA, inhibiting the latter from diminishing APC's inactivation of factor VA. The inactivation of factor VIIa is not as well understood. The half-life of factor VIIa is only around 2 minutes unless factor EPCR is present to stabilize it. Some have questioned the significance of APC's inactivation of factor VIIa, and it is unknown to what degree factor V and protein S are cofactors in its proteolysis. It is known that APC works on factor VIIa by cleaving at 2 sites, ARG336 and ARG562, either of which is sufficient to disable factor VIIa and convert it to factor VI. Cytoprotective effects. When APC is bound to EPCR, it performs a number of important cytoprotective functions, most of which are known to require EPCR and PAR1. These include regulating gene expression, anti-inflammatory effects, anti-apoptotic effects and protecting endothelial barrier function. Treatment of cells with APC demonstrates that its gene expression modulation effectively controls major pathways for inflammatory and apoptotic behavior. There are about 20 genes that are upregulated by protein C, and 20 genes that are downregulated. The former are generally anti-inflammatory and anti-apoptotic pathways, while the latter tend to be pro-inflammatory and pro-apoptotic. APC's mechanisms for altering gene expression profiles are not well understood, but it is believed that they at least partly involve an inhibitory effect on transcription factor activity. Important proteins that APC upregulates include BCL2, eNOS and IAP. APC affects significant downregulation of p53 and BAX. APC has anti-inflammatory effects on endothelial cells and leukocytes. APC affects endothelial cells by inhibiting inflammatory mediator releasing downregulating vascular adhesion molecules. This reduces leukocyte adhesion and infiltration into tissues, while also limiting damage to underlying tissue. APC supports endothelial barrier function and reduces chemotaxis. APC inhibits the release of inflammatory response mediators in leukocytes as well as endothelial cells, by reducing cytokine response, and may be diminishing systemic inflammatory response, such as is seen in sepsis. Studies on both rats and humans have demonstrated that APC reduces endotoxin-induced pulmonary injury and inflammation. Scientists recognize activated proteinace's anti-apoptotic effects, but are unclear as to the exact mechanisms by which apoptosis is inhibited. It is known that APC is neuroprotective. APC's anti-apoptotic effects are part of the reason that APC is effective in treating sepsis, as reduced levels of apoptosis are correlated with higher survival rates in septic patients. Anti-apoptosis is achieved with diminished activation of caspase 3 and caspase 8, improved BAX-BCL2 ratio and downregulation of p53. Activated proteinace also provides much protection of endothelial barrier function. Endothelial barrier breakdown, and the corresponding increase in endothelial permeability, are associated with swelling, hypertension and inflammation, all problems of sepsis. APC protects endothelial barrier function by inducing PAR1-dependent sphingosine kinase 1 activation and upregulating sphingosine 1-phosphate with sphingosine kinase. Role in disease: agenetic proteinace deficiency, in its mild form associated with simple heterozygosity, causes a significantly increased risk of venous thrombosis in adults. If a fetus is homozygous or compound heterozygous for the deficiency, there may be a presentation of purpurefulminans, severe disseminated intravascular coagulation and simultaneous venous thromboembolism in the womb. This is very severe and usually fatal. Deletion of the proteinace gene in mice causes fetal death around the time of birth. Fetal mice with no proteinace develop normally at first, but experience severe bleeding, coagulopathy, deposition of fibrin and necrosis of the liver. The frequency of proteinace deficiency among asymptomatic individuals is between 1 in 200 and 1 in 500. In contrast, significant symptoms of the deficiency are detectable in 1 in 20,000 individuals. No racial nor ethnic biases have been detected. Activated proteinace resistance occurs when APC is unable to perform its functions. This disease has similar symptoms to proteinace deficiency. The most common mutation leading to activated proteinace resistance among Caucasians is at the cleavage site in factor V for APC. There, ARG506 is replaced with GLN, producing factor V Leiden. This mutation is also called R506Q. The mutation leading to the loss of this cleavage site actually stops APC from effectively inactivating both factor VA and factor VIIIa. Thus, the person's blood clots too readily, and he is perpetually at an increased risk for thromboses. Individuals heterozygous for the factor Leiden mutation carry a risk of venous thrombosis 5-7 times higher than in the general population. Homozygous subjects have a risk 80 times higher. This mutation is also the most common hereditary risk for venous thrombosis among Caucasians. Around 5% of APC resistance are not associated with the above mutation and factor Leiden. Other genetic mutations cause APC resistance, but none to the extent that factor Leiden does. These mutations include various other versions of factor V, spontaneous generation of autoantibodies targeting factor V, and dysfunction of any of APC's cofactors. Also, Some acquired conditions may reduce the efficacy of APC in performing its anticoagulative functions. Studies suggest that between 20% and 60% of thrombophilic patients suffer from some form of APC resistance. Warfarin necrosis is an acquired protein C deficiency due to treatment with warfarin, which is a vitamin K antagonist and an anticoagulant itself. However, warfarin treatment may produce paradoxical skin lesions similar to those seen in purpura fulminans. A variant of this response presents as venous limb gangrene when warfarin is used to treat deep vein thrombosis associated with cancer. In these situations, warfarin may be restarted at a low dosage to ensure that the protein C deficiency does not present before the vitamin K coagulation factors II, IX and X are suppressed. Activated protein C cleaves Plasmodium falciparum histones which are released during infection. Cleavage of these histones eliminates their pro-inflammatory effects. Role in medicine. rhAPC has been the subject of significant controversy since its approval for clinical use in 2001. A 2011 Cochrane review concluded that it does not decrease mortality in severe sepsis or septic shock. It has been noted that rates of severe hemorrhages, drug infusion-related fatal events and termination of infusion due to adverse reactions are all higher in clinical use and open-label trials than in controlled trials. There is a dispute as to whether or not studies after PROC confirm its results, and if so, for what subgroups. Protein C levels have long been noted to predict mortality in patients with sepsis. Because of this, and its pleiotropic anticoagulative and cytoprotective effects, protein C has long been suggested, along with many other drugs, for use in treating patients with severe sepsis. In November of that year, the Food and Drug Administration approved drotocogan alpha activated in the clinical treatment of adults suffering from severe sepsis and with a high risk of death. Drotocogan alpha activated is a recombinant form of human activated protein C, that is, it is a protein produced by recombinant DNA. It is marketed as Xedris by Eli Lilly and Company, but recently recalled and taken off the market. APC has been studied as way of treating lung injury, after studies showed that in patients with lung injury, reduced APC levels in specific parts of the lungs correlated with worse outcomes. APC also has been considered for use in improving patient outcome in cases of ischemic stroke, a medical emergency in which arterial blockage deprives a region of brain of oxygen, causing tissue death. Promising studies suggest that APC could be coupled with the only currently approved treatment, tissue plasminogen activator, to protect the brain from TPA's very harmful side effects, in addition to preventing cell death from lack of oxygen. Clinical use of APC has also been proposed for improving the outcome of pancreatic islet transplantation in treating type I diabetes. Notes: AI plus or minus a GLA domainless protein C is produced by selective proteolysis between residues 82 and 83 to remove the N-terminal portion of the protein that includes essentially all of the GLA domain. The N-terminus was removed in order to make crystallization of the protein easier. AI squared and hypereosinophilia, excess eosinophil-specific granule proteins on the endothelial surface bind to thrombomodulin and inhibit its participation in the activation of protein C by electrostatic interaction on the surface of thrombomodulin. References. External links: Office of the Commissioner. Xedris, drotocogan alpha, March 2005. Retrieved September 16, 2010. Nifin, Cassandra L. Protein C. OMIM. Retrieved 2010-09-13. PDB entry 1 ot. PDBE. European Bioinformatics Institute. Retrieved September 13, 2010. PROC Protein C, Homo sapiens. Intrasgene September 9, 2010. Retrieved September 13, 2010. The MEROPS online database for peptidases and their inhibitors, S1.218, the protein C pathway. John H. Griffin, TSRI, La Jolla, California. Diagram of the blood coagulation pathway and protein C pathway. Product information: human protein Euro, mouse protein Euro, bovine protein C. Hematologic Technologies Incorporated. Retrieved September 14, 2010. Product information: human activated protein EPCR, bovine activated protein EPCR, mouse activated protein C. Hematologic Technologies Incorporated 2009. Retrieved September 28, 2010.